Okay, we're going to talk about metabolism in the body. I introduced it on Tuesday. We're going to start here. So, we're talking about nutrients moving from the gut, liver, circulation through the heart, and then to where they're needed in the body. Once those nutrients arrive, they can be used to build or burn, as the case may be. So, when we think about a cow and what a cow does, we need to think about a cow as metabolism. That's their big strength, the ability to process nutrients and generate an output, in this case, milk production, that far exceeds most other animals and what their abilities are. So we've selected these animals, we've bred these animals. So let's get a feel for where parts of metabolism occur in the body and how things move through the body. Again, we're looking at liver as the center. So we're going to be using our friends here again. We've got F for fat prep, G for glycolysis, P for protein prep, T for transition, and O for oxidation. So we have all the potential pathways on the left side of the screen here. We have glycerol coming out of the triglyceride, going to go in through glycolysis, transition oxidation. The fatty acids will be split into two carbon pieces. They'll go directly to oxidation. Carbohydrates, GTO. Protein will be broken down into the amino acids. The amino acids will have their ammonia group removed, and the carbon skeleton, that keto acid, will either go PGTO, PTO, or PO to be metabolized to CO2 and water. So that's what we talked about on Tuesday. We're going to represent this diagram over here on the left this way in the following diagrams. So kind of simplifying that shorthand. We've already mentioned the three carbon units. We're going to be showing you where they fit into metabolism. Remember, we've got propionic acid, lactic acid, pyruvic acid, glycerol, and alanine. And for the purposes of this class, they can be very interchangeable. They all have that three carbon backbone there, three carbons in a row, and that's where the interchangeability in this class comes from. So the three carbon units, what we want to think about is the breaking apart of glucose, which we've discussed already in glycolysis. We have the six carbon ring here of glucose. We don't have the extra oxygens and hydrogens on it, just the backbone and the closing of the ring with the oxygen. We run it through glycolysis. We get two three carbon units, which are called pyruvate. Now we can run this backwards, and this is very important in a cow. We take two three carbon units, we put it through gluconeogenesis, or I'll abbreviate it GNG, and we can recreate, with the addition of ATP here, we can recreate that glucose. And we saw all the different three carbon units that we can have. Any of those coming in can be used to recreate glucose, and this is rather important in a dairy cow, and we'll show you why. So we can provide the cow with three carbon units. One of the things that we'll do to give that cow is propylene glycol. It's a three carbon backbone. As it gets absorbed, it gets taken to the liver and can become part of that glucogenic cycle, taking its three carbon structure, taking two of them together, and creating a glucose. So this is the way we used to do this class. We kind of go through, I build this step by step, drawing things out, and you would draw along with me, and that's how we taught it. Given our online status, this is going to be not something we can do. So what I've had to do is convert this into a computer format. It's taken longer than I hoped it would, but I think I've created something here we can use to help you understand metabolism in the cow. So this is the same thing in a more computerized format. This is all that we're going to learn here. There's a lot on this slide, so it's going to take a while to break it down and walk us through. You can either print out this slide in the lecture handouts. This will have everything, and you can follow along as we build it up. Or you can start with this blank one and using the color key up here in the corner, black, green, blue, red, you can fill things in as I discussed them along the way. So this is the new version here. I think I've got everything in that and created in such a way. So we've already talked about this online, but for those of you that weren't there, we have the body represented here. We got our eye and our nose of our cow, the year we're doing, and we have the circulatory system here. Here's the venous side of the circulatory system, ending in the vena cava, coming back to the heart. The heart pumps out a cycle through the lungs, and then we come back and then pumps it out the aorta. The aorta provides blood to the GI tract, here represented by the reticulorumen in the forestomach and the small intestine, the connection between those. We have the portal vein that's going to connect the gastrointestinal tract in the liver. The liver is going to be supplied also blood directly from the aorta, and that'll circulate blood, capillary bed, veins, venules going back through. We've got the adipose tissue, and there's a circulation associated with that. We've got the muscle, we're going around, nutrients moving around. Nervous tissue, we're going to look at that as a more terminal. Things that go there, especially for creating energy, do not leave. So stuff goes there, does not come back out. Mammary gland is going to synthesize milk, and that's going to be exported from the body, so that's taking it out of the circulation. We're not going to have circulation through there again. So this is going to represent our body, and we'll fill it up with metabolism to teach you what's going on. So we'll start sort of how the digestive tract brings things in. So again, blue is processes, metabolic pathways, green is nutrients. So rumen reticulum, we have fermentation, carbohydrates, NDF, neutral detergent fiber, non-structural carbohydrate, starches, sugars are going to be fermented, and they're going to be turned into VFAs. The three most common VFAs are acetate, which is 2 carbons, propionate, 3 carbons, and butyrate, which is 4 carbons. That fermentation goes along. It's going to digest some of the feed. It's not going to touch other portions of the feed. Whatever leaves that fermentation vat is going to move down to the small intestine. This is where the bulk of our digestion and absorption occurs. The microbes that were running the fermentation are going to be used as a protein source. That protein source will be digested down to amino acids, so microbial amino acids, the AA there. Those amino acids will be absorbed and available to the body. We have feed that wasn't fermented. Those proteins can be broken down into individual amino acids, and that also becomes part of the absorbed amino acid pool. We're going to have a certain amount of starch and a certain amount of possibly glucose that gets by the rumen. More often, it's the starch that's broken down into glucose, and then the glucose is absorbed. We're also going to have a limited amount of fat. There's not a lot of fat in a dairy cattle diet. That will be absorbed to the small intestine. One of the unique features about fats is they don't go to the liver first. They get absorbed into the lymph system, which runs parallel to the venous system, and they get dumped into the circulation here, and they bypass the liver. So they're going to go out to the body first. In this case, more often than not, to the adipose tissue and the muscles, and hopefully not too much back to the liver. So you have this here. Again, everything that's available, everything that's absorbed is going to go through the portal vein and end up in the liver. So let's think about the metabolism that occurs in the liver. We talked about the liver as kind of the crossroads of metabolism. You're going to have the ability to turn fats, carbohydrates, and proteins into energy, getting them down to oxidation. We talked about synthesis and degradation. We're going to focus on protein synthesis and degradation. So we take amino acids, we synthesize protein, the chains therein. We take that protein, we break it down, and we have amino acids that we can recycle and use for other proteins. We also have amino acid synthesis in the liver. So the non essential amino acids are created in the liver and they provide amino acids to the pool that the liver has to work with. The other main process we're going to talk about is taking 3 carbon units and turning them into 6 carbon units. Gluconeogenesis, we've got the key down here if you need it. We take our 3 carbon units, we run glycolysis basically backwards, and we generate glucose. Those glucose units can be run together in chains, and glycogen can be created. This is the storage form of glucose. You're going to find it in the liver and also in the muscles where it can be there. So, given all the processing that occurs in the liver, what's going to leave the liver more often than not is acetate. So, that's from the VFAs up here. We'll get to that more in detail. Glucose from our glucose pool here, amino acids from our amino acid pool, protein, and then urea if the liver is ripping apart amino acids. It's going to have to take the ammonia groups that it rips off and it'll turn it into urea, and then urea is exported out around the body. So let's take a look at where some of the VFAs end up. So let's start with acetate and butyrate. Um, Acetate has 2 primary fates. The 2 carbon can feed directly into oxidation and provide energy for the liver. And that is the preferred substrate that the liver in a cow will use to generate energy. That acetate can also not be absorbed, not be utilized, and pass out of the liver into the circulatory system and then become available throughout the body for other tasks. Butyrate seems to be the preferred fuel for the rumen and the reticulum, omasum and abomasum, as their energy source. So we have butyrate, 4 carbon. It's going to go through fat prep and its beta oxidation. It's going to be cut in half. So two carbon pieces, which feed directly into oxidation. So a limited amount of butyrate gets through. Most of it will use to be running the GI tract. Um, if it does, it can usually pass through and be absorbed or used in other areas of the body with that fat prep oxidation in other parts of the body. So those are even numbered VFAs, 2 carbon acetate, 4 carbon butyrate. We're going to talk about propionate. Propionate's a 3 carbon unit. It gets absorbed across the reticulum wall, or in the omasum, ends up in the portal blood. Almost all that propionate, that 3 carbon unit, is going to go into gluconeogenesis. So 3 carbon units enter here, glycolysis runs backwards, gluconeogenesis, and we create glucose. There's very little, and we'll show you in a bit here, there's very little raw glucose absorbed in a cow's diet. in order for her to generate milk components that we talked about on Tuesday, she's going to need glucose, and the only way that gets done is with propionate. So propionate is the primary source of glucose in the dairy cow. That glucose is exported and then it's taken around the body and used where it's needed. You're talking in a dairy cow, you're talking 5-10 pounds of propionate is converted to glucose on a daily basis. So that this pathway in the cow's body, a lactating cow producing over 100 pounds, very important. So we've got protein synthesis and degradation. You've got uh, carbohydrate degradation, carbohydrate synthesis, and fat degradation, all parts of the liver's metabolism. So depending on where things go, um, we can have different fates as to what we're going. But again, the liver is kind of the crossroads of metabolism. If we focus on our amino acid pool, so we have from the gut, we have amino acids from the microbes or amino acids from the feed. They are absorbed, the proteins are broken down to amino acids. They are absorbed and they go into the amino acid pool. They can either be used to synthesize protein, they can pass out of the liver on to help protein synthesis in other parts of the body. Alanine, a non-essential amino acid, one of these amino acids that you're going to find the 20 there, is a 3-carbon unit that can pass directly into gluconeogenesis. Alanine can also be synthesized as a non-essential amino acid in the liver, but alanine can provide a source of 3-carbon units. Those 3-carbon units can be used to generate glucose. So the last thing we'll talk about here being absorbed is the starch. The starch is broken down into glucose. So whatever starch doesn't get fermented to these fatty acids will pass to the duodenum. Enzymes will come out, break it down into its glucoses. When glucose is absorbed across the lining, it generally goes through glycolysis. So that's going to generate pyruvate and then lactic acid. Lactic acid can move in the blood out of the cell and into the bloodstream. That now becomes a substrate for gluconeogenesis. Lactate is another carbon unit um, that can be used there. Anywhere it occurs in the diet, the body can come into the liver and also be added to this pool for that. So I'm not really sure why starch glucose is broken down. I guess it's a supplement to the butyrate, but that lactate moves on to it's a glycogenic precursor. So we're making the glucose with that lactate. So if we look at all the potential things that could feed into um, our gluconeogenesis, where can the 3-carbon units come from? So we said lactate is one that either can come from the gut as glucose is burned going across or glycolysized or whatever. You can have lactic acid build up in the muscle that can circulate through, go around, come in this way, and then be one of the 3-carbon units there. We already mentioned alanine. In theory, alanine can be absorbed from the gut or may come from this pool over here and add to that 3-carbon units. We said propionate, that's a big one. That's the majority of the glucose is made from propionate. We can have the glycerol from the breakdown of fat going into the 3-carbon units here, or we can take pyruvate at the end of glycolysis, and that can also provide a 3-carbon unit. And then we can add the various substrates that we might supplement a cow when she needs some help with her metabolism, and we'll get to that in 2 lectures. So this is pretty much all the nutrients and what they do. So acetate can either be burned in the liver, pass through. Propionate, it's all going to go through gluconeogenesis. Butyrate is burned before it gets to the liver. Amino acids are absorbed and they're part of the pool that can go in a lot of different directions. Glucose usually is absorbed as lactic acid that goes back into gluconeogenesis, and our fats bypass. So what goes into the liver, what comes out? Acetate, glucose, amino acids, protein, and urea are the substrates that are leaving. From liver metabolism, as you go around, we have triglycerides or free fatty acids. Fatty acids, they're sometimes called NEFAs, N-E-F-E, non-esterified fatty acids. Acetate, amino acids, protein, glucose, water, minerals, and vitamins. These are all the things that will be distributed through the body to help the body do what it needs to do. So what happens in adipose tissue? So we have these nutrients here available. The adipose tissue, the fat tissue, all over the body. So let's focus on what's on our back. Will absorb pre-made fats. So we've got these fats here. They'll be brought in and stored in some way, shape, or form. Or it'll take the building blocks of glucose and acetate and create, through the process called lipogenesis, the triglyceride. So the glucose will be broken in half by glycolysis. We will take those 2 pyruvates and turn them into glycerol. They are going to be the hanger on our triglyceride. Acetate, they'll be strung together, 2 carbon units be strung together 2 at a time, and those are going to become the fatty acids that hang off that glycerol. So we take these building blocks, we go through lipogenesis, we've got triglycerides. Breaking those triglycerides apart, that stored pool of triglycerides, the process, we break stuff down again, primarily to glycerol and fatty acids. We'll call that lipolysis. That is promoted by epinephrine. So if you get a good scare, you're going to have your fats pulled out of storage and made available for energy metabolism. So if you get a good scare, that's part of where the buzz comes from, is your body saying that you need to do some stuff. You need energy to get things going. So adipose tissue, creation of triglycerides, the breaking down of triglycerides. Main purpose is storage of fats for later use. So these are the things that go into the fat. What comes out is triglycerides that are put in the carriers that float by. The carriers are made in the liver. We have the protein carriers with their hydrophobic on the inside, and our fats and will go inside that, go into the circulation, and go around to the body and dropped off wherever they're needed. Looking at muscles, we have amino acids coming in. There's a synthesis there, and we create the proteins that are necessary for muscle contraction. So the actin myosin complex, actin helix, those proteins can also be broken down and regenerating that amino acid pool. So we've got the build up, the synthesis of protein, and the degradation of protein that occurs in our mammary gland. So again, blue. DEG is degradation. SYN is synthesis. We have metabolism here. You're not going to burn a lot of protein in the muscles. You're going to focus on the fats. That'll go through fat prep, and then whatever routes we need to do to get to oxidation. We have glucose coming in. It can be stored as glycogen in the muscle, but then we break it down to individual glucoses that can go through glycolysis, transition, oxidation to generate ATP. So we need oxygen present for complete oxidation. If there happens to be no oxygen present, no O2, we can run glycolysis, but we can't run oxidation. As we mentioned before, oxygen is required for the full burn, and we don't have that. We're going to get through glycolysis, and we're going to have pyruvates that we can't do anything with. So what happens is pyruvate is not a good thing to have around in the cells. So the cells create lactic acid from the pyruvate. So one 3-carbon unit is turned into another 3-carbon unit. The thing about lactic acid is it can be exported from the cell. Pyruvate won't leave the cell. Lactic acid can. So lactic acid gets exported and recycled through the body. So it's going to go all the way around and probably end up in gluconeogenesis in the liver. As protein amino acids are absorbed, we recycle, go through protein cycles. We can also export amino acids from the muscle going through. So you've got the nutrients going in. We're showing amino acids here, fats, glucose, and then what we export is amino acids and lactic acid or lactate. As we mentioned on Tuesday, the nervous tissue tends to prefer glucose as a substrate, and that glucose is used to burn GTO, glycolysis, transition, oxidation, to generate the energy that the nervous tissue or the brain needs. So that glucose goes in, burned completely as CO2 and water. Nerve cells tend to stay. They don't turn over. They don't break down that much. So generally what goes in is energy once everything is built, and that energy is generated through those 3 processes. We can have these nutrients also going to the mammary gland. So mammary gland can take all these things here and they can make milk, and that milk is exported from the body to feed a calf, to feed us humans, to make cheese, ice cream, all the things that we love. So hormones, they are appearing in red. We already mentioned epinephrine will promote lipolysis or lipolysis, breakdown of triglycerides. So if you get scared, we need energy. We're going to break that fat down. We're going to make it available for the burning of energy. BST, growth hormone, bovine somatotropin, is used as a lactogenic promoter. That BST is secreted from the hypothalamus, pituitary gland. That moves to the liver. It's converted to IGF, or starts an IGF cascade. IGF leaves the liver. It's going to circulate around and it's going to end up in the mammary gland. It's going to promote milk production. So that's how BST, through that secondary messenger, promotes milk production. The other one we want to mention here is insulin. Insulin puts things away. So if we've got extra components in the blood, we want to put them away. So we think about insulin is primarily about glucose. So insulin is going to promote glycogen synthesis in the liver. but more directly, it's going to start glycogen synthesis in the muscle. The muscle is much more responsive to insulin than the liver is. So amino acids or glucose is going to be glycogen in the muscle, and it's also going to move the glucose into other cells. You're going to have amino acids. Their uptake is promoted by insulin. And then you're going to have triglycerides, NEFAs, free fatty acids are going to be stored. The fats, the promotion of storage. So we're saving the energy for a later time or putting it away for later use in one way, shape, or form. So that's how some of the key hormones work. So that's metabolism in our cow. So we used to did our milk production this way. This is the way we're presenting it this year, given what we've got. We talked a little bit about how we get across in lab last week. I will spend more time on that in another lecture. This one's here in this slide as well. So same netted thing, just a different kind of format of presentation. More linear approach than the circular approach that we just did. Here's the same thing for 2019. The things I want you to think about related to metabolism, and we'll talk about this as a review next week. Where does acetate go? So it's produced in the rumen fermentation. Where does it go? So how does it get used in the body? Where is it burnt? Where is it used to build? Same thing with propionate, butyrate, and microbial amino acids or any amino acids. They get absorbed. What happens to them? What are the potential fates for each of those items? So give that some thought. Take time with those schematics and trace where those compounds, those nutrients might end up and what their ultimate fate is. So that's sort of taking what's out of the gut and then